Okay, let's move on to part 3, common cancer terms. We've already introduced some terms throughout this lecture, but now let's kind of go over the ones we've talked about and also introduce some others that we haven't touched on yet. So, a neoplasm, neo meaning new, and plasm, as we've seen, means growth or formation, means new tissue. And so neoplasm is actually the formal word for a tumor. Now, tumors can either be benign, so a benign tumor is not spreading and not considered cancerous. So benign growths can occur where the cells have some changes or mechanisms that are possibly dysregulated, causing some increased cell growth, but they're not yet cancerous. So some benign growths never even become cancerous, but it's important to keep an eye on them and observe them to ensure that they don't evolve into a cancerous growth. And malignant means that these cells are dysregulated, not as tightly contained, meaning badness, meaning to be born. So these cells are not as tightly contained of the cancer type and can spread into neighboring tissues. So malignant tumors can become metastatic. That's where we see the spread, right? So metastatic, again, meta meaning change, stasis meaning placement, so spreading to other parts of the body. So metastatic is referring to that spread. And cachexia, coming from Greek kakos, meaning bad, poor, harmful, hexia, meaning condition, state of being. Cachexia refers to a rapid wasting of body tissue. This is observed in cancer patients, primarily from not eating or from some of the treatments. And so cachexia involves a extreme loss of body tissue, so muscle mass, all thinning of the skin, so rapid wasting of body tissues. So some additional terms referring to types of cancers. So carcinomas are the most common type of cancer. These are formed specifically by epithelial cells, which form linings over the body's exterior and also on interior surfaces of the body. So lung, for example, breast, colon, bladder, and prostate. And again, carcin, as we've said before, carcino actually has a root, or as a combining form, has Greek origin meaning crab, and this actually is derived from the fact that the shape of the tumor is kind of being hard and round, if you will, and we know that the suffix oma means tumor. So carcinomas are the most common type. It's just, as we saw in that photo of the epithelial cells growing to form a solid mass or tumor. Sarcoma, so sarco is the combining form meaning fleshy. So sarcomas are tumors that form in bone and soft tissues, such as muscle, fat, even blood vessels, so fibrous tissues. Leukemia does not form tumors, but with leukemia, you get uncontrolled cell growth of blood cells. So in cases of leukemia, your blood cell count will be elevated, which can be seen on blood work. So in leukemia, we don't see tumors necessarily, but instead blood cell count becomes altered. And so leuco, the combining form, means white or colorless. And as we've seen previously in last week, emia refers to a condition of the blood. A lymphoma, or lymphomas, are cancer cells of white blood cells and lymphocytes, so cells of the immune system. Lymph, referring to the lymphatic system, lympho, as a combining form, clear water, which refers to lymphatic fluid. So lymphatic fluid, the fluid that runs through our lymphatic system and through our lymph vessels, is a clear fluid. So lymphomas are cancers of the lymphocytes, so cells of lymph nodes are within the lymphatic fluid. Multiple myeloma is cancer of plasma cells. So plasma cells are a type of immune cell that produce our antibodies. So plasma cells are responsible, they become specialized as antibodies. And multiple myeloma forms tumors in bones. And the origin here, myelo means marrow, or referring to bone marrow. And finally, melanoma is cancer of melanocytes. So if mutations occur in melanocytes, which are the cells that form skin pigment, mutations that occur here can cause abnormal growth of skin cells and form a skin growth. And so the skin cells which produce melanin is the pigment of these cells. The combining form melano represents dark pigment. And so if you get mutations in these cells, you may end up having melanoma, which is cancer of these skin cells. Okay, so for naming cancers, I just wanted to point something out here. As we talked about in last lecture, the notation that we use to represent combining forms, we have the root and then we use a slash with an O, right, to show that combining form vowel. So instead of how it's represented here as adeno with a dash beside it, this is just how it was used from the image from this site, but I just wanted to clarify that the proper notation that we will be using as well is written as adeno or chondro with the slash in between in our combining form. But this photo nicely shows the different cancer combining forms. So adeno is referring to gland, so chondro referring to cartilage, erythro, so again, if there's cancer of the blood, as we saw before, or the erythrocytes, erythro refers to red blood cells, hepato, liver, lipo is fat, lymphocytes, hence lymphoma was tumor of or cancer of the lymphocytes. Here we see that melano, so melanoma for pigment cells, myeloma for bone marrow, osteo for bone, so osteoma would be a tumor of the bone. Okay, so when a tumor is found, there's a standardized grading system, and it has to do with tumor size, whether there's lymph node involvement, so if the cancer cells have spread or metastasized from the original location that it was found into the lymphatic system, there might be some lymph node involvement, and also metastasis, so if it has traveled to other parts of the body or not. So based on the size, if it's in the lymph nodes and if it has spread, there is a particular grading system of being graded as, 0 would be not detectable, and then 1 would be on the lower end, 4 would be malignant. So 4 is being the highest grade or the worst. So you don't need to know, I will say, the specifics of each of the 3 facets here of the T and M grading or the scaling here, but just to be aware of these 3 features here, right? So size is important. Typically larger tends to be associated with, further cell growth. Lymph node status, swelling of lymph nodes, so lymph nodes feeling swollen or lumpy, can be a sign that there is some cancer there, status showing that the lymph node is inflamed or enlarged, and metastasis, right? So whether the lymph nodes in the surrounding area are cancer-free or not. So some cancers use this TMN screening. Some others have different staging systems, but just to introduce this to you guys, more important here is the stages. So benign cancers, or something that's benign, would maybe sit in stage 0. If it gets to stage 1, the cancer is relatively small and contained and so forth. But as we increase in stage, sometimes in stage 2, you might see some spread into lymph nodes that are close to the tumor. But then in stage 3, we can start to see perhaps some spread to surrounding tissues. And then finally in stage 4, the cancer has metastasized and moved well beyond where the origin was. So now let's look at just general causes of cancer. We've talked about what increases risk factors, and there are many different things, and often these things are specific to type, so to the specific type of cancer. Underlying genetic factors can increase your risk of developing certain cancers, so a higher predisposition of specific cancers that are inherited. Environmental factors are a big one, so radiation, some specific types of viral infections, so HPV, or having been infected with HPV, is associated with an increased risk of cervical cancer, and Epstein-Barr virus, or EBV, is associated with a higher risk of developing lymphoma. We'll talk about Epstein-Barr virus more in a little bit. But chemicals, toxins, referred to as carcinogens. A carcinogen is just a chemical that is mutagenic, which means that it can cause mutations in our cells. So there are known chemicals or toxins that are known to be carcinogenic and are able to alter the structure or influence our DNA to cause mutations. And lifestyle choices, we already talked about how diet, weight is associated with cancer risk, particularly after menopause, and certain types of cancer, such as breast cancer. Exercise has been shown through research to influence risk of developing cancer, as exercise can help prevent certain types of cancers or reduce the risk, if you will. Smoking, alcohol use, as we talked about. I'm going to again these two terms here again. So carcinogen, carcino meaning cancer, gen meaning producing. So this is a chemical that is known to increase the chances of developing cancer. And a mutagen is a substance that causes mutations to DNA. So Epstein-Barr virus is a very common herpes virus that actually infects most people, often in childhood. Usually upon infection, it causes mild or no symptoms, but it is famous for causing mononucleosis virus that you may have heard of in teens and young adults as well, and it's very commonly spread through saliva. So once infected, EBV stays in your body for life in a dormant state, but can reactivate. And so research has shown that Epstein-Barr virus is actually linked to certain cancers and autoimmune conditions, even like multiple sclerosis. But it has been shown that being previously exposed to or having had Epstein-Barr virus is actually associated with a higher risk of developing certain types of cancers. So it can cause many illnesses, including lymphoma, which we talked about, malignant tumors in the nasopharynx, so tumors in the back of the nose and throat, and also immune deficiency. So just having had a prior exposure to this virus, this is one of the risks. So in introducing you to these, for example, Epstein-Barr virus and these things, we're just trying to bring awareness to the fact that as modern medicine has expanded and as research has been conducted on the underlying risk factors or underlying causes of certain cancers, we're better understanding these various risks, and therefore this information can help us to create more targeted treatments or by better understanding the source or potential risk factors for these given cancers. And so as we saw that trend of, that reduced overall mortality risk that we're seeing upon incidence of cancers. So all of this greater knowledge on cancer cells and cancer development is really helpful for that type of research. Okay, so interestingly, this is also related to EBV, but interestingly, most of the population has been infected by EBV. So we can actually test if someone has antibodies for the virus. The presence of antibodies in your blood will show whether you've had that virus or not. So this graph shows different age ranges on the bottom, and then it shows the percent of individuals with antibodies in their blood. So a high percentage of people have this antibody. Doesn't mean that you're going to get cancer for having had this virus. It's just to note that certain viruses, or there are certain risk factors that we can actually measure that are associated with certain types of cancers. And again, certain aspects of our environment will also increase the risk. So this information, again, it provides insight into potential areas where we can maybe find and develop treatments and so forth. Now, let's go over some of those screening techniques and warning signs, because we have evolved in science and research to recognize signs that may indicate cancer's presence. So, something to note, again, not all of these, observations mean that an individual does have cancer, but these are signs that have been observed in people who have developed a certain cancer, for example. So changes in bowel or bladder habits may be associated with bowel cancer or bladder cancer. A sore that does not heal, unusual bleeding or discharge, unexplained weight loss or appetite, difficulty swallowing, which is dysphagia, so difficulty swallowing, new mass or changes in the breast. Some other naming systems like caution, again, change in bowel habits, sores that don't heal, nagging cough or hoarseness. So we've developed kind of these signs to look out for, and Canadian Cancer Society and so forth. But again, noting correlation does not always equal causation. These are just some measures and screening tools that are shared in this regard. Now, childhood cancer warning signs look a little bit different. They tend to present slightly different and are specific to the type of cancer that may be in effect. Things like, white spots in the eyes, a new squint, lumps, of course, any place on the body, but mostly the stomach, glands, groin area, unexplained fever, aching, neurological signs. But the key here is that childhood cancer may present slightly differently than it may in adults. So some screening tests. So screening can be used to rule out if something is cancerous or not. Skin cancer screening method is a very common measure for skin moles and to determine if a mole is cancerous. So they use this ABCDE protocol here. So A, asymmetry. So if a mole is asymmetrical, as you can see here on the left, this mole is very round. This one has very jagged edges. And so border is next, blurry or jagged edges. That can be a sign that the mole may be cancerous. If there's more than one shade or color, this can point to maybe a cancerous mole. Size, diameter, it's larger or growing, but the biggest thing is evolution. And so changes over time are the most important indicator if a mole is cancerous. Typically any one of these on their own or any even two of them on their own don't necessarily point to a mole that's cancerous. It's typically that evolution. If you're seeing growth and changes in the border, becoming more jagged, evolution is kind of the key here for skin cancer screening. So there are many procedures that can be performed to screen for cancer, and these are procedures that are regularly performed, many of them after reaching a certain age where we know the risk of developing a certain cancer is higher. For example, a colonoscopy. So breaking down that word, again, as we saw last week, scopy means to look, colon, referring to the colon. This is a screening of the large intestine for tumors and polyps. This allows for early detection. Many times a polyp can be discovered and removed at the time before it can develop into anything dangerous. And so colonoscopies, for example, have significantly reduced the mortality rate of cancers of the colon because we have this screening measure where we can provide that early detection that we talked about. Mammogram, mammo means breast and gram means a record. So this is actually taking a record of the breast tissue. You can see that sometimes a very small lump in the breast can be difficult to detect or feel at all using a breast exam. And so a mammogram is going to provide a very detailed photo of the breast tissue to be able to visualize any lumps that may be present. A Pap test is actually, it's kind of funny. So Pap is not actually a medical term necessarily. This test was named after the individual who created the test. His last name was Papanicolaou, along the lines of that. So they named it the Pap test. But the Pap test is collecting a uterine cell sample. So collecting cells from this region and being able to determine if the cells have any malignant changes. Prostate exam, in order to feel and examine the prostate and the size of the prostate, determine if it's enlarged, and a biopsy. So again, bio meaning life, opsy, sight or to see. A biopsy involves taking a small sample of a tissue, tumor, mole, so forth, for further examination to determine if the cells are displaying any signs of cancerous activity. we see, it's interesting, so why screening is so important, kind of what I want you to take away from this is using prostate cancer as an example here. So if you look at 1,000 men with or without symptoms, out of 1,000 men that did not, without screening, so say screening for prostate cancer did not exist, out of 1,000 men, 68 will be diagnosed with prostate cancer, 7 will die of prostate cancer, and 61 will be treated and will survive their cancer. Now, that's without any screening measures. With screening, so taking those proactive measures, out of 1,000 men, 88 will be diagnosed. And so we're increasing the number who will be diagnosed, right? Because we're screening, we're detecting. So we're catching it early and more. 7 will still die, but 81 will be treated and survive. So more are being treated, there's more detection, and we are able to, prevent things from kind of developing into something a little bit harder to treat. So screening, very important and is really helpful in reducing that mortality rate that we were looking at. Some people may have done this. Program 23andMe can give you feedback on developing certain diseases. Some people don't care to know this information. This just showcasing another modern technology that has been developed for screening. So something like 23andMe allows you to screen for your disease risk. So your genetic risk of developing a certain disease or certain type of cancer. For some, perhaps interested in positively informing you of any lifestyle changes that you may want to make to help with reducing the risk further. Now, this slide was added by my colleague to say that while there is screening we can do or precautions we can take in life to be aware and learn more about things that are natural and good for us and prevent disease, it's interesting to note, and the key of that, moderation is key. So, for example, this is going over lethal doses of common chemicals. And interestingly, water in very, very high doses is actually considered toxic. And having too high levels of water can actually be unhealthy and can cause negative consequences on the body. Interestingly, things like caffeine, something that some of us consume every day, is actually considered moderately toxic. The term here, LD50, is the lethal dose of common chemicals. So highly toxic ones are those that are toxic in very low doses, but basically to say moderation is key, of course. We've recognized a lot of, risk factors or mutagens or carcinogens, certain things being cancerous, certain things not, but also to say that any chemical, moderation is key and not trying to go to extremes, but extremes of something is not necessarily a good thing. So the last thing we're going to talk about is HeLa cells. So this is a development and research that is quite interestingly and is actually still very widely used today in research and continuously around the world, helping with the study of very many different things. So HeLa cells are a widely used human cell line in laboratories. So basically, in 1951, the cervical cells were harvested from an individual who had cervical cancer. And these cells were taken into a lab to monitor their changes, so to monitor their division, to observe them. And the researchers noted that these human cells could survive and keep dividing indefinitely outside of the body. So essentially an immortal cell line. Now, this is really helpful from a research standpoint because we could better understand cancer biology and the dynamics of cancer cells. And they allowed us to study genetics, cell biology, and then these cells could be used and treated with different chemicals and toxins to be able to determine how certain chemicals influence our cells. So HeLa cells have been used for a very long time in research, and we've also developed other various cell lines over time, but these were really the first cells that were harvested and grown in a laboratory to be able to study cell biology and cancer biology and so forth. So a really, really interesting and helpful development in biology. And for those of you who are interested, I don't expect you to know all this information, really just knowing what HeLa cells are. But if you are interested in learning more about HeLa cells, I have included this slide for you, as it's a very interesting development in scientific research. Okay, so to summarize, I've included a summary here just highlighting some of the key themes that we've talked about in this lecture. Biggest takeaways, kind of what is cancer? What is the biological basis of cancer cells? What genes are involved in the development of cancer? Cancer has a major societal impact, right? And we talked about different terms like potential years of life lost, incidence and mortality rates. We talked about some of those hallmarks of cancer, so including evading apoptosis, sustained cell division or proliferation, angiogenesis, being able to gain a nutrient blood supply, and metastasis or spreading. We talked about the stages of cancer progression, risk factors, and then we also talked about classification, screening, prevention, and early detection, and all of the advances in screening and treatment that have led to those improved survival rates that we saw in the graphs at the beginning of this lecture series. So that wraps up our talk on our cancer unit. So if you guys have any questions, feel free to email me at any time. A reminder that your worksheet opens up by the time I'm posting that will be considered today. So it'll be posted on Tuesday, January 20th. It'll be available under assessments quizzes. So it's a quiz style type of worksheet activity. You can pause and come back to it. So save your work and come back as you work on the worksheet. And you have until Friday at 5 p.m. to complete it and submit it. So I will talk to you next week. Take care.